Please, your.
Sir, is it visible?
Respected chairman and co-chairman, it is visible.
Yeah, it's visible. Make it in full screen.
Okay, Mr. Madhudiam, please proceed.
Good evening, one and all. Today I am here going to present my oral presentation on trade competitiveness and evolving export patterns of Indian marine products, a decadal analysis. So, introduction. So, India is the one of the important oceanographic and with abundant water resources in inland and marine sector. It facilitates the captured and marine fishes. So India is the one of the largest fish producing country in the world, with shares about 9.12% to the global fish production. Indian fisheries sector is a vital component of the country's economy, play a crucial role in ensuring the food security for the nation. And India emerged as a global leader in the fish sector that contributes 1% to the GDP and 6% to the agriculture GDP. And first in inland captured fish production and cultured shrimp, and second largest aquaculture producer globally, and fourth largest exporter of marine fish products, and sixth largest producer of the marine captured fish. So the major importing markets for Indian marine products were 20% major importing markets are Middle East with constitute about 20%, followed by the European Union. Where in case coming to the next graph, the major supplying markets for the fish and crustaceans, mollusks, and other aquatic invertebrates imported by India. The major importing from the Bangladesh, which accounts about 24%, followed by the USA, Vietnam, and Myanmar, and so on. So the research question is to study the export competitiveness and trade direction of Indian marine products in the global market. And research gap, no studies have been taken up comprehensively covering all the major marine products and their export competitiveness. And this research provides the insights into the marine products to ensure improved global competitiveness. Subject to see the export competitiveness and the methodology criteria of export product and to analyze the trade. So comparative is the reporting that, and to analyze the revealed symmetry comparative advantage, RSCA was used to analyze to overcome the asymmetry in the revealed comparative advantage. So it is calculated by RCA minus 1 divided by RCA plus 1. And the Markov chain analysis was used to analyze the trade direction of the major marine products. And the fish production in fish production trends and growth in the world. So here we can observe from the from 2002 to 2023, a steady increase in the production with a CAGR of 1.89% at 1% level of significance. And the fish production trends and growth in India. Here we can observe that the total fish production, it was increase. The total fish production has steadily increased from the 6,399,000 tons to in 2002 to 2003 to 4 to from 17,544,000 tons in 2022, reflecting a compound annual growth rate of 5.63% per annum at 1% level of significance. And in case of marine fish and inland fish production, which is significant positive increasing growth rate with 1% level of significance. And the trends in growth and instability of major marine products in the different time periods, the both in terms of quantity and value, it has shown a positive growth rate at the 1% level of significant. And in case of instability index, we can observe in terms of value, we can observe the low instability in the variation in the export of that particular total marine product, where in case of in terms of value in the period overall period, it can observe that high instability. And growth and instability of export of marine products or different products, here we can observe that highest positive growth rate in dried items with 1% level of significant, followed by the frozen fish. In terms of value, we can observe from frozen fish due to increase in the monetary value, and followed by the dried items. In case of instability index, we can observe that only dried items and chilled items were observed high instability compared to other marine products. So competitiveness of Indian marine products export, here we can observe that the different marine products were shown. Only among that only 3 products are comparative advantage in exporting those products, which are frozen fish and crustaceans and mollusks, and all other marine products are showing comparative disadvantage in exporting these particular products. Coming to trade direction of major marine products export from India, the trade direction analysis is fundamental technique used to understand the evolving the pattern of trade between the countries or trading countries. And here in the transitional probability matrix, the diagonal cells represent the likelihood of a corresponding importer remaining in the same trade state or in India. The values in the row indicates the probability of losing the trade relationship to the competing importers, while those in the column indicates the probability of gaining the trade due to competition from the other countries. So the transitional probability matrix of Indian fish, fresh and chilled export from the India to the world. So among the key destinations, Bangladesh, Nepal, Bhutan, and so on. The Bhutan is the major, the most stable importer of Indian fresh chilled and marine products, with retention percent of 93% original share from the previous year, and it lost 7% to the China and gained from 4% from the Nepal. And the most unstable values we can observe from the diagonal elements, which are showing 0, which indicates most unstable markets to export the marine products, that particular product to those countries. Similarly, in case of crustaceans, here we can observe that the most stable markets are USA, followed by Vietnam, and in case of unstable markets are UAE, Canada, and Russia. In case of frozen fish, here we can observe that the China is the most stable market, and followed by Thailand, and most unstable markets are Tunisia, Malaysia, and Bangladesh, and so on.
Madhu DM, please conclude. Your time is over.
Yes, sir. The last table. Here we can observe from the table the transitional probability items, the quantity of exports of marine products from 2013 to 2022. So it is evident from the table that the frozen shrimp is the most stable marine product export, as a probability of frozen shrimp retained its original share from the previous year was about 82%. It lost their share 6% to the dried items and 10% to the 10% and gained from 93% from the drained items. The most unstable export marine products among the major exported products were frozen squid, dried items, and live items with 0% of retention capability. So coming to the conclusion, the Indian fisheries sector has a significant role in the economy, providing nutritional food security, generating income to the export earning, and offering the employment for more than 2.8 crore people in India. There is a significant growth in the export value of 14% and quantity of 7% of the Indian marine product between 2003 to 2022 across the various categories. However, the growth of both quantity and value is unstable in the indicated by instability index. Hence, the sustainable export growth. It is important to implement the policies that enable the diversifying the export markets and investing the value added processing and improving the supply chain. Thank you, sir.
Thank you, Madhu DM. Now it is open for discussion. Respected chairman and co-chairman, please, if you have any queries, please ask to Madhu DM. Thank you, sir. Hello.
Hello, sir.
Hello. Yes, sir.
Hello, sir.
Please, sir, please.
DM Singh, sir.
Yeah. What is the basic constraints of marketing drawback in our country?
Sir, sanitary and phytosanitary measures were the problem, and some countries have some we can getting some residual amount in the marine fish product due to the pollution in the ocean. So that lots of the marine products was rejected in the previous years. So the sanitary and phytosanitary measures need to be followed by exporters of in our country, sir.
Okay.
Hello, Madhu.
Yes, sir.
As per your presentation,
Yes, sir.
India ranks first in cultured shrimp,
Yes, sir.
but we are lagging in export. What may be the reason?
Sir, we are the fourth largest exporter, sir, because fourth largest exporter in the world, sir.
Yes, yes.
So even though India has a large population, the domestic requirement also required, sir.
Yes, yes.
So that for huge population after a domestic requirement, the India is exporting the surplus quantity of the marine products to other countries, sir. And even though some kind of processed products also exporting from India, sir.
In present consequences,
Yes.
this one tariff means US is we have given relaxation for 90 days.
Sir?
If we are going for reciprocal tariff
Yes, sir.
barriers, what will be the implications? What will be the implications on our export?
Sir, we need to follow the sanitary and phytosanitary measures, sir. The requirement of the demand from the other countries, whatever their requirement, we need to give those kind of suitable products to those countries, sir.
No, that is already we are following these things.
Yes, sir.
I am asking about this one reciprocal tariff. US declared reciprocal tariff
Yes, sir.
on
Sir,
our export.
sorry, I don't know about it, sir.
Okay, it's okay.
Okay, sir. Thank you, sir.
And transitional probability matrix.
Yes, sir.
You have used this one for comparing our neighboring country or whatever export we are exporting.
It is, sir, for some particular product. For example, in case of crustaceans, here we can observe that in the row, in the diagonal elements, which indicates that retention from the previous year. Sir, for example, 75% of retention from the previous year. That means for every year, the India is exporting 75% to the crustaceans to the USA. In case
Yes.
of in the row, other countries indicating that that percent of remaining 25% loss to those countries, sir.
Okay.
So in case of in the column lines, the USA is gaining from the other countries, sir.
Actually, USA is now going to impose reciprocal tariff.
Yes, sir.
Reciprocal tariff means they are going to increase the tariff on our export.
Yes, sir.
So they have given 90 days relaxation.
Yes, sir.
After 90 days, they are going to implement these things. Now they are putting 45% tariff on Chinese goods
Yes, sir.
and products.
Okay, sir.
Anyway, thank you. Nice presentation.
Thank you, sir. Thank you, sir.
Okay, thank you, Madhudiyam.
Thank you, sir.
You can leave now. And this is the final call for the poster presentation. Mrs. Pooja Gill, are you there?
Yes,
No.
sir.
So please proceed.
Sir, I will say.
Yeah.
Sir, can you see?
Yeah.
Hello.
It is
Hello.
visible.
Yes, yes.
Good evening, everyone. Myself, Pooja Gill, MVC student in veterinary pathology, CVS Navania. Today, I am going to present a case of chronic nephropathy in the Asiatic elephant. Chronic nephropathy is a long-term disease of kidney where the kidney tissue slowly get damaged over time. It leads to a gradual loss of the kidney functions, which can eventually cause of a kidney failure. Damage can cause the tubular degeneration, glomerular congestion, and fibrosis. Chronic nephropathy can happen due to the old age of and any infections, toxins, and low blood supplies or any immune-related problems. This is a postmortem report of a 50 years old Asiatic elephant. That is postmortem conduct in the Animal Husbandry Department, Udaipur. In the first picture, we can see the highly hemorrhagic spleen and the discoloration of the spleen also. In the second picture is the part of the liver that having the necrotic foci and hemorrhages, multiple hemorrhages also. In the third picture is showing the cross sections of the heart and is having the hemorrhages and the congestion also. And the fourth picture is showing the part of the kidney. It's having a lot of the hemorrhages. And the picture A is showing the histopathology of the kidney, that is showing the chronic interstitial nephritis with the diffuse interstitial lymphocyte infiltration and the associate with the fibrosis and tubular atrophy. That blue color dots denoting the lymphocyte infiltration and the all over the blue color discoloration in the first A picture is showing the fibrosis. The picture B is showing the second B is showing the hemorrhages around the tubules. The hemorrhages, the red or pinkish, dark pinkish dots are showing the hemorrhages. And the picture C is the showing the collapsed alveoli and the emphysema. The collapsed alveoli, we can see the alveoli look shrunken and the flattened. And there may be the crowding of the alveoli walls. They look almost touching each other's walls. And the picture D is showing the spleen tissue look darker overall because of the brownish pigment hemosiderin discoloration. And the hemosiderin is the iron storage pigment. Because of that one, it's showing the more darker and brownish color. Picture E is showing the periportal necrosis. The periportal necrosis, death of the liver cells around the portal tire. And in the normal structure, the liver cords near the portal tire is in this picture. The liver cord cells are lost. Picture F is showing the kidney epithelial cast. The cast look like a pink solid cylindric filled with the cells inside the renal tubules. The conclusion and the result, the case is highlight the significance of the age-related degenerative changes and their impact on the vital organ function of the genetic elephants. Thank you, sir.
Thank you. Thank you, Pooja Gill. Now it is open for discussion. Respected chairman and co-chairman.
What was the major cause of death of elephant?
Sir, elephant's age is around 50 years. So because of the age related, all changes are because of the age related, sir.
Okay. So is any other abnormalities you have found in post-mortem examination?
No, sir, no other major abnormalities. All are the age related, like the hemosiderin pigmentation on the liver, that also because of the age related only, and the hemorrhages and the this one nephritis also because of the age related.
Okay.
Can we go for next presentation?
Okay, sir. Thank you, Pooja Gill. Next presentation, oral presentation, Dr. Sharlene. Dr. Shirley, are you here?
Yes, sir.
Please proceed.
Am I audible, sir?
Yes, yes. Go ahead.
Her name is not there in list.
With your permission, Dr. Dinesh sir has suggested, sir.
Okay.
Three candidates were missing in the list, sir.
So, a very good evening, sir. So today I'm here.
Actually. Okay, go ahead.
So today I'm here to present my research work on nutritional status of fishes from the parent stream of River Ganga, a contribution towards food and nutrition security. So, as we all know that fishes are the most important source of nutrition for human population. So, the main different constituents of the fishes includes proteins, carbohydrates, lipids, as well as minerals. And also among the mineral elements, there are micro elements as well as micro elements. So human body usually need all the kinds of the nutrients for the proper health, as well as the deficiencies these nutrients can cause a variety of the diseases in the human beings. So our study is basically restricted to the Alaknanda River. So this Alaknanda River is basically the parent stream of the River Ganga. So on this River Ganga, we basically chose 6 different cyprinid fish species like Schizothorax richardsoni, Schizothorax plagiostomus, Schizothorax progastus, Berlius bendalensis, Crossocilus latius, and Gahira lempta. So these were the objectives of our research work. First objective was the total moisture and total protein content estimation in the muscle tissue of selected fish species. Second objective was total lipid and total carbohydrate content. Third objective was the concentration of mineral elements. Fourth objective to compare the nutritional status of selected fish species. So on basically we chose two different sites on the river Alaknanda. One site was located on this that is in the Srinagar district, and another site was located in the Tilwara district. So another one is the methodology, and in the methodology, we basically proceed with first step, that is the collection of the fish species. So 6 different fish species were collected from the 2 different sites, and they were carried in fresh condition to the laboratory to find out its nutritional value. Then in the laboratory, the fish species were identified with the standard keys and subjected to sample preparation. So these were the fish species basically selected for the study. So this one is the methodology: collection, identification, and chemical analysis. So chemical analysis is basically in that chemical analysis, we analyze total protein content according to Lowry method, total lipid content according to Paul's method, total carbohydrate content according to Du Bois, and mineral and moisture content according to the AOAC 2000. So these were basically the results. So this table is showing the different 6 cyprinid species that were collected during the study period, common name, and their mean length and weight, along with the feeding habits. So of all the fish species, Richardsoni and Plagiostomus were herbivorous in nature. Crossocilus latius, Gahira lempta were herbivorous as well as detritivoric. Another species like Schizothorax progastus and Berlius bendalensis, they were omnivorous in nature. So this one table is basically showing the seasonal variation in the total moisture content among the 6 cyprinid fish species. So in all these 6 species, maximum moisture content was recorded during the monsoon, while minimum was recorded during the winter season. Another one is this one is the seasonal variation in the total protein content of 6 dependent species, and here the maximum protein content was recorded in the summer season, followed by the monsoon and then winter season. Next one is the seasonal variation in the percent lipid content in all the species. So in this, maximum moisture content was recorded during the monsoon season, followed by the summer and winter season. So this one is the average biochemical composition, which is found in the muscle tissue of all the selected species. So in this, the moisture component is basically the dominant component, and followed by the protein, lipid, and carbohydrate content. And as per the classification of Rahman et al. 1995, we found all the different species that we selected. These are basically low fat fish species, as they contain the lipid, approximate 1 to 5% lipid content. Next one, this one is the figure which is showing the total moisture content among the different species. So Richardsoni, first of all, that is the Plagiostomus, is basically having the maximum moisture content, and minimum content was recorded in the Crossocilus latius. So in the protein, with respect to the protein content, Gahira lambda is found rich in the protein content, and very minimum amount of protein content was recorded in the Plagiostomus. So as far as the lipid component is considered, so lipid content was found maximum in the Borrelia bendalensis, and almost the minimum and almost equal amount was observed in Richardsoni, Plagiostomus, and Progastus. Next one is the carbohydrate content. So minimum carbohydrate content was recorded in the Plagiostomus, while maximum was reported in the Crossocilus latius. Next one is the mineral composition. So a total of 3 macro elements were selected for the present study. So of these macro elements, sodium, calcium, and potassium were analyzed in all the selected species. So this one, sodium, calcium, and potassium were found maximum during the monsoon season, and followed by the summer and the winter season. Next one is this one is the average variation of macro elements in the muscle tissue of all the studied species. So in this, we can observe the maximum, that is abundance pattern of the macro elements, as calcium is the dominant element, followed by the potassium and then sodium. So sodium forms the least component with respect to the macro elements. So this one, sodium component or sodium element was found to be maximum in Gahira lambda, while minimum in case of the Plagiostomus. So another component is basically the calcium, and calcium was found maximum in Borrelia bendalensis, while minimum in Schizophractis progastus, and potassium content was found to be highest in Crossocilus latius, and minimum in case of the Borrelia bendalensis. So this one is the trace element, and three trace elements were selected for the study, and one heavy metal is selected for the study in order to find out the toxicity in the fish. So iron, zinc, and lead and copper were analyzed in all the fish species muscle tissue. So as far as the iron content is considered, so iron was found to be maximum during the monsoon season, and it is basically followed by summer and winter season. So winter season forms where in the winter season very less concentration of iron was reported in all the studied species. Next one is the zinc. So maximum zinc content was recorded during the winter season, followed by summer and monsoon season. And lead content was basically recorded maximum in the summer, followed by winter and monsoon season. Copper, which is the last trace element we have analyzed, and this one copper is found highest during the summer season, followed by monsoon and winter season. So this one table is showing the average variation of the different trace elements and one heavy metal in all the studied species. So this one abundance pattern is basically in the muscle tissue of all the studied fish species, iron forms the dominant element, followed by zinc, copper, and lead. And also this one lead content is compared with the food and agriculture with the permissible limits given by FAO and WHO, and it was found that this lead content is very less than the permissible limit as set by these standard organizations. Next one, iron content. Iron content was found to be maximum or highest in case of progastus and followed by other species, and minimum content was recorded in Barilis bendalensis. As far as zinc content is considered, so zinc content was found maximum in Barilis bendalensis while minimum in Plagiostomus. So lead content was found maximum in Barilis bendalensis and minimum was recorded in Plagiostomus. So last one was the trace element, which is the copper, and copper was found minimum in Plagiostomus while maximum concentration was accumulated in the Schizothoracis progastus. So another is the conclusion. So all these studied Himalayan fish species were basically were of high nutritional value in terms of all the in terms of total protein, total lipid, macro, as well as trace elements. And trace element concentration like copper concentration, iron concentration, zinc, as well as lead concentration also were found below the highest permissible limit as set by FAO and also WHO. So hence these fishes can be considered as safe for human consumption.
Thank you, Dr. Shirley. Now it is open for discussion. Respected chairman, if you have any question, and respected co-chairman, please proceed. Go for your first slide. Dr. Shirley?
Yes, sir. This one?
This. Well,
Yes.
okay. Sir, please.
DN Singh, sir.
Yeah. As Dr. Charlie Sharma, you have not correlated or comparative study of the nutritional value of Himalayan fishes and Ganga fishes.
Other fish species. Sir, actually we have published a paper in which we have given the comparative account of a species with other type of the species, but here it is not mentioned.
Because you have mentioned the nutritional status of fishes from the parent stream of river Ganga.
Yes,
And
sir, actually a wide variety of species are there, but we have chosen approximate 7 species. Out of the 7 I have presented here is 6 species. So we compared basically the different parameters like biochemical and trace elements in all the studied species in this slide, basically.
okay. Which method is being used for a study of the micromineral examination of the
yes, sir. That basically we have estimated by using AAS, atomic absorption spectrophotometer, and with the help of double digestion. First of all, we have digested the samples. Then samples were then diluted up to 100 ml. Then basically that sample we have sent to the Anacon laboratories for this one find out the concentration of minerals, because in our laboratory we don't have that AAS.
Okay.
Sarali, Dr.
Yes, sir.
Sarali. Actually, my concern is that your objective, you have given 4 objectives.
Yes, sir.
It's better to give 2, 3 means if you are putting proximate analysis, you will be limit 1, 2 in one objective. Anyway,
Okay, sir.
you put GSI, gonadosomatic index, so that you can compare a better way muscle means how it is seasonally changing. So you can better reflect your work.
Oh, okay,
And calcium, potassium, sodium
जी.
content in muscle is varying from season to season. What may
Yes,
be the
sir.
reason?
Sir, maximum was basically found in a monsoon. So maybe there are the different agricultural fields also. So maybe the elements are basically coming from that and directly entering into the water body. Because at the along the bank of the river, there are basically different villages and agricultural fields are also there. So maybe the source of the elements will be from the agriculture field and this.
Even iron is also high in summer season.
Monsoon, sir,
Yeah,
followed by
monsoon.
yes.
I think it will be less in case of monsoon.
Okay, sir, but we have found this pattern. Less we have found with respect to the zinc and lead.
The 65.
Actually, you do one more parameter,
जी, जी.
blood plasma composition for same nutrients,
Elements. Okay, okay, sir.
so that you can compare that this may be utilized for gonadal development.
जी, जी, sir.
So
Okay, sir, we will
anyway,
take
it's
care of
good.
it.
And in case of pollution, you have studied PB. But
Yes, sir.
in Himalayan region, there is, I
Yes.
think, least pollution. So
Yes, sir.
it
No
reflected less.
industries, no factories are there, so very
Yes,
less
yes.
amount of.
It's okay. Nice presentation. Thank you.
Thank you so
Okay.
much, sir.
Thank you, Dr. Shirley. You can leave.
Thank you, sir.
Next oral presentation, Dr. Jasmeet. Are you ready?
Yes, sir.
Okay. Dr. Shirley, please leave. So
Yes,
that
sir.
Dr. Jasmeet can share the slide. Dr. Jasmeet, please share your slide. Make
Yes,
it
sir.
fast.
ये कहाँ?
Okay, go ahead.
Yeah.
Good evening to one and all. I'll be talking on a bit different topic from the theme which is running down in the session. That is airborne transmission and antimicrobial resistance genes of E. coli in zoos of Chhattisgarh. I would like to focus that everyone is talking about one health, one health, one health. This one health starts from your soil, plant, animal, fish, everything, environment. So whatever contaminants or heavy metal in soil, it comes to
Dr.
plant
Jasmeet, make it a
like
full
that.
slide.
Sir, I will finish it. Don't worry, sir. So we know we have lot of bacteria which are resistant to bacteria into environment, mainly from animal husbandry. And this E. coli bacteria is a very effective indicator for prevalence of these resistance studies in different ecosystems. So I have isolated these organisms from air and dust samples from different zoos. No one has attempted this one in our country till now. And I have identified the multiple drug-resistant isolates, and I've also done the presence study of presence of LAT, MSHV, and CTXM genes in these. So a total of 30 air samples and 30 dust samples from 3 different zoos of Bihari, Bilaspur, and Raichur were collected. So a total of 180 samples were collected. So this is a protocol. For dust, we used a sterile brush, collecting it directly into a sterile container. And for dust sample, keeping a EMB agar plate in an open area, 1 meter away from any obstruction, 1 meter height for 1 hour. Then subjecting it to isolation procedure where the pinkish colony on MLA agar followed by typical metallic green sheath which confirms it to be phenotypically E. coli was done. So all of these isolates were also molecularly confirmed to gene level by 16S RNA gene, followed by antibiotic sensitivity test using the following antibiotic tests. And again, the phenotypic and genotypic also a detection of blood, TEM, and SHV and CTXM genes was done. So on the basis of results, we could confirm that whatever we phenotypically screened were molecularly confirmed to be E. coli. When coming to antibiogram assay, most common drugs which we are using, like moxiclav and tetracycline, even in animal husbandry practices, the moxiclav was almost 93.13% resistance, while tetracycline was 83% resistance. Then coming to susceptible drugs, you can see the advanced drugs, carbapenem group, imipenem and azotrinom, which are rarest used in animals, were found. Similarly, you
Dr. Jasmeet, your voice. Dr. Jasmeet? Hello?
He has disconnected.
He got disconnected, sir. He's not disconnected. Okay. We lost his
Sound
connection.
problem. Maybe sound problem.
No, Dinesh sir, he's left actually. His connection is lost.
We can proceed for next.
Okay.
Dr.
Next is
Lipika.
Dr. Aarti Gupta.
Yes,
Are you
sir.
ready?
Yes, sir.
Okay. Proceed, please.
Is it visible, sir?
Yes.
This is poster presentation.
Yes, sir. Good evening, everyone. Myself, Aarti Gupta. I'm a BSc scholar from CBS Navania. My presentation is on effect of Nigeria sativa black cumin on blood biochemistry of Kadaknath birds. The research trial was carried out on 200-day-old Kadaknath chicks that was purchased from a certified commercial farm in Udaipur. Chicks were randomly divided into 5 groups, T1 to T5, 40 chicks in each groups, and 4 replicates. In T1, it's a control group where no Nigeria sativa is given. In T2 to T5, diet with 1%, 2%, 3 and 4% Nigeria sativa is given respectively. All groups were raised on deep litter flooring with the standard measurement, management, hygiene, vaccination protocol. Feed and water given is ad libitum. On day 42, blood was collected from wing vein, 3 ml each, 4 birds per group. In EDTA treated sample, we analyzed RBC, WBC, hemoglobin, PCV, heterophils, lymphocyte, and their ratio. We use serum for analyzing cholesterol, glucose, triglycerides. Through this table, we can see that in T1 group, T1 is control group, T2, T3, T4, and T5. In T5 group, RBC, PCV, hemoglobin is highest because black cumin have antioxidant property. A active ingredient known as thymoquinone, which is active compound, a strong antioxidant. It protects RBC from oxidative damage caused by free radicals. When RBCs are healthier and live longer, there is number of RBC naturally increase. So RBC carry hemoglobin. That more RBC means more hemoglobin and better PCV. It also stimulates blood cell protection, stimulate bone marrow activity where blood cells are produced. Some study also suggest that black cumin enhance enhanced production of erythropoietin, a hormone that is responsible for RBC formation. Higher RBC count, higher PCV, and higher hemoglobin. It also having anti-inflammatory effect. A chronic inflammation can suppress blood cell production. Anti-inflammatory properties decrease the inflammation in body, which help the bone marrow focus on producing more healthy blood cells, better nutrient absorption from gut, so more absorption of iron, vitamin B12, and folic acid and other nutrients are critical for RBC formation. So these RBC, PCV, and hemoglobin increases with given black cumin. And heterophils, when we give black cumin, there is less tissue damage because it is having an antioxidant property, so less inflammation, so less heterophils in number. Black cumin also kills or inhibit the bacterial growth, so less infection, overall less WBC count, especially heterophils that is seen in table first, WBC and heterophils. It also works as immunomodulation. Black cumin doesn't blindly stimulate the immune system. It also balances it, boost protective immunity like antibody production, but also calm down unnecessary cellular immune response. It keeps immune system efficient without exhausting it. In second table, we can see that glucose is increasing in T5 group because it also affects pancreas, insulin. So glucose is converted into glycogen. So in T5 group, highest amount of Nigeria is given, then lower blood glucose. It also inhibits cholesterol synthesis in liver. Thymogen, which is antioxidant, also block the enzyme HMG coenzyme reductase, which is responsible for making cholesterol inside the liver. Less enzymatic activity, so less cholesterol production. It also improve lipid mobilization, so it help to remove LDL, which is bad cholesterol, and triglyceride from the blood and help to increase the LDL. So as a conclusion, we can say that dietary inclusion of Nigeria sativa improve the hematological parameter and reduce serum cholesterol, triglyceride, glucose in Kadaknath birds. And 4% inclusion level showed that more pronounced benefit, indicating a strong hematic and lipolipidemic potential of black cumin with dose-dependent effects. Thank you, sir.
Thank you, Dr. Aarti. What is the active ingredient in black cumin?
Sir, thymoquinone.
And approximate composition of black cumin is?
Sir, it's a protein is approximately 20%
Are you sure?
CP is approximately 20, moisture content is 6.8 almost, nitrogenous free extract is 19%, and crude fiber is 6.3%, and ash is 3.8.
Okay. So how you decide the level of inclusion? Why not 5%, 10%?
Sir, its maximum limit is 2 to 5%. So 2 to 4%. So we divided groups as to maximum limit.
From where you get 2 to 5% maximum level? Are you conducted experiment earlier?
No, sir.
Then how?
Sir, we referred it from other previous papers.
Okay, then you are also within that limit only, using black cumin, that limit only up to 4%.
Yes, sir.
Okay. Beyond that, any adverse effect you see?
No, sir.
Okay. Okay, thank you. If anyone want to ask, then most welcome. Chairman sir and co-chairman sir.
Yeah, Dr. Aarti, you have not mentioned about the economics of black cumin feeding in Kadaknag.
Yes, sir.
So how can you suggest this experiment is beneficial for our farming community?
Sir, it's cheap, but I don't include it in this poster.
Okay. What is the tentative cost?
We are using in our kitchen also. It's black cumin. It's most common.
Yeah. It should be mentioned.
Okay, sir.
It is
Okay, thank
around 30 to 40 rupees per 100 gram, I think.
₹30 to 40 rupees per 100 gram.
Yes, sir.
Oh.
Respected co-chairman, would you like to ask any question?
The
Then, sir?
presentation is very clear. It's okay. If any other presentation, we can move ahead.
Yes, sir. Okay, thank you, Dr. Aarti. You may leave.
Okay, sir.
Dr. Lipika Sharma. Hello,
Yes, sir.
Dr.
Yes,
Lipika
sir.
Sharma. Please proceed. Make it
Okay,
fast.
sir. Thank you so much, sir. So please allow me to share the slides, sir.
We have already allowed.
Thank you, sir.
Yes, it is visible.
Sir, 1 second, sir. I'm not getting the file, sir.
Dr. Lipika. Dr. Lipika. I think so. She lost her connection.
She lost the connection.
Yes, sir.
Anyone
Okay.
remaining to present?
Dr. Sunil.
Yes, sir, available.
Dr. Sunil.
Sir, available, sir.
Make it fast. Please proceed.
Sir, my screen is available, visible, sir.
Yeah, yes.
Yeah, sir, this is Sunil Gaude. Actually, I'm working as a manager in the microbiology. So, my abstract related to the endotoxin assessment and controlling the ketoprofen injection for safety first approach. Introduction. So, what is the ketoprofen? Ketoprofen is a widely used non-steroidal anti-inflammatory drug. So, openly administered via the injection to treat the pain and inflammation. So, why endotoxin is matter? Endotoxin derived from the gram-negative bacteria pose the significant hazards when introduced parentally. So, need to control. It is very important. And it's also regulatory landscape like international standards like the USP 85, European guideline 2.6.14, JP 4.01, and IP 2.2.3. So, aim of this study. Hello. My screen is visible, sir. Hello.
Yeah.
Dr. Sunil,
Yeah,
please
visible,
be
sir.
on your presentation.
Yes. Aim of this study. The study aims to develop the strategic approach for the detecting and qualifying and controlling the endotogenic ketopropanization during the manufacturer proactive endotoxin management, emphasizing the proactive science-based
Dr.
approach.
Sunil, we are not able to see your slide. Be on your presentation, on your PDF, what you are sharing. Do not
I am
move here and
presenting
here.
only, sir. Madam, sir, so one call came just right. Okay, I'm on. Actually, I'm in the company, so I'm waiting from last 2 to 3 hours. So it is mobile is not means camera is not allowed. So I am trying from my mobile. Just wait. Is visible, sir? And validation
Yes, good.
Yeah, validation methodology employ validated techniques for the reliable endotoxin detection and control. So method we are used here to bacterial endotoxin test. This is a protocol. We are using here gel plot method for the detection of the endotoxin. First, this ketoprofen injection, which is not available, limit is not available in the any pharmacopoeia. So I selected this product. So I have calculated this first. If you are checking or detecting any endotoxin from the any parental product, we need to check first. We need to calculate endotoxin limit first. So based on limit, we can develop the protocols and validations. So I have calculated it based on the K by M. Is the K is the maximum acceptable limit per kg body of the weight, and M is the maximum daily dose of the test product per kg of the body mass. So yeah, for generally as per the guideline, is the 5 U per kg is our those product we are injecting the intravenously or intravenously. So M is a 20 mg per 70 kg is a ketoprofen body mass based on the taken from the tag insert of the respective product, medical product. So is after calculation, endotoxin limit is calculation is coming as 1.725 U per mg. So for the dilution, we to calculate MVD. So the MVD is the maximum valid dilution. So it is based on the endotoxin limit into concentration of the product or potency of the sample divided by the license sensitivity. So MVD means we calculated 1.75 U per mg is a ketoprofen limit into 50 mg per ml is the product concentration divided by 0.03 U per ml is a license sensitivity. So calculation is coming as 2912. So based on this dilution, means it is a MVD means we can dilute the product up to the 2912 m is up to 2912 ml. So here table 3 is mentioned that I calculated, I have diluted the product up to the MVD is the lower highest the concentration versus lower dilution. So MVD by 2, then MVD by 16, MVD by 8, MVD by 4, MVD by 2. I calculated base and you can see there dilution degrees concentration. Once we dilute it, means MVD is the maximum value dilution, where the product concentration is very less. So once you detect the very concentration of the product at a lower dilution and low higher concentration, so that we can give that limit to the any product, means you can give that product limit to the any step of the during the manufacturing. So these are the gel plot method. So method suitability tests are crucial to confirm this test. So we did here. So for the MUD calculation, and first of all, you have to do the sample composition preparation. So 50 mg per ml is the concentration. I have taken 3 batches, different batches for the data collection. Sample presentation involving the MUD by 32 to the MUD. Then multiple batches are tested to ensure the consistency and reliability of the endotoxin detection. So these are the methodology. So validated our validation approach. First, establish the NID, means non-inhibited dilution of the product. Then same non-inhibited.
Conclude.
Yeah. Okay. So based on this, this validation, the sample was prepared up to the dilution up to the 32 MUD by 32, and this was established as a non-interfering dilution to the MUD by 2. The test dilution demonstrating no inhibition enhancement, confirming the compatibility of the LAL reagent. Based on the validation outcome, the routine BET analysis is conducted using the test sample prepared at MUD by 4 and tested at MUD by 2. And the validation approach enables detection of the control and endotoxin in the betapropane injection up to the 0.342 mg per ml concentration. This is significantly more stringent than the calculated endotoxin limit, that is 1.75 U per mg, demonstrating robust and sensitive method. Hence, this method ensures the reliable endotoxin detection and contribution to the safety and regulatory compliance of the injectable product. And based on this limit, we can control our entire manufacturing process.
Dr. Sunil, please show your conclusion.
Sir.
Okay. Now it is open for discussion. Respected chairman and co-chairman, please.
Which method is being used for detection of endotoxin level?
Sir, endotoxin, we have LAL reagent, sir, LAL test. Limulus amiboside, limuloside, lysate. 50-50 method we use.
Okay.
Yes. Gel clot method.
Yeah. Is it efficient method? What is the accuracy?
Accuracy means we can quantify the quantification only. Means if our product limit is 0.25 U per mg, we can detect the endotoxin less than 25 U per mg. Means it is not giving it exact, but it is giving a less. Means whatever the limit we have given to the product. But it is a cost effective because of this lysate is coming around 1 vial 5,000 ml. But if we are going for the PTA method, it will cost around PTS also is a rapid method, but cost around 125,000, 31 lakh up to. So it is a cost effective method.
Okay.
So, thank you, sir. We will proceed for the next presentation. Lipika Sharma, Dr. Lipika Sharma.
Yes, sir.
Thank you.
Present, sir.
You may leave, Sunil.
Yes.
Please. Dr. Lipika Sharma, please proceed.
Sure. Okay, sir.
Ensure your internet connectivity.
A very good evening to all the present here. I am Dr. Lipika Sharma. Today I am going to present a poster presentation, and the topic of the presentation is comparative analysis of thermal adaptability among indigenous and crossbred cattle through biochemical and gene expression markers. As the global temperature is rising, so the heat stress is impacting the dairy farmer at a huge scale. It hampers the production and the productivity of the animals. Ultimately, it creating a huge loss to the nation's economy, as well as it is creating problem in the food security. most of the farmers, small and marginals, are depend on their production and productivity of their animal. So it is very important to find out a solution for the heat stress affecting animals, at the same time to maintain the productivity of animals. Indigenous, for example, Haryana, they are considered as more thermotolerant than the crossbred cattle. Nowadays, government also giving importance to improve the quality of the indigenous cow, as they are to be conserved as they are more thermotolerant and they are more resistant to diseases. So the conservation of the indigenous breeds is very important. So with that objective, the gene expression on during and for these studies, 5 Haryana and Vindavani, which is a crossbreed, was considered, and the animals of between 1.5 to 2 years, and the animals maintain in a cytotic chamber at 38 and 40 consumed. So weeks from PM to 2 PM, and some were collected animals, and blood test was done for the to see the expression pattern of the genes such as HSP70, HSP90, interleukin 2, interleukin 6, endothelial nitric oxide synthesis. Cortisol was higher in crossbred animal during acute heat stress, but as the heat stress progressed, it took the chronic phase. Then the cortisol level was was in the normal range. It shows that the cortisol at the very beginning during acute heat stress, it was higher because of the adaptability mechanism of the animal. As the days increases, because of the negative control, negative feedback mechanism, the animals adaptability towards the heat stress increases, and hypothalamo-pituitary axis starts decreasing the concentration of the cortisol in the blood. So here we see that the level of cortisol, which is a stress marker, is already considered was higher in case of Vrindavani. And coming to the second graph, we see the HSP70, which is a heat shock protein gene. The expression was higher in case of Vrindavani cattle. HSP70, they actually they are responsible for the maintenance of the protein structure. The stress animal generally they show the higher expression level of the HSP70. So here we see that in case of Vrindavani cattle, the heat shock protein 70 was found higher, and in case of HSP 90, the level of the HSP 90 was found higher in case of indigenous cattle, that is Haryana. It showed that how Haryana cattle maintain its thermoadaptability adaptation mechanism in both acute and chronic heat stress. And the interleukins are mainly are the cytokines, which shows that how the immune system react to the heat stress of the animal. So interleukin 2, which is considered as the pro-inflammatory cytokines, it was shown higher in case of Vrindavani in order to cope up with the higher ambient temperature. The cells tries to release higher level of interleukin in order to cope up with the all the infectious diseases, or they try to release the T cells, which will be which are actually helpful for animals' disease resistance capacity. And we see the relative mRNA expression of the enos, which is the endothelial nitric oxide synthesis, and this actually involved in the conversion of L-arginine to nitric oxide in the blood, and nitric oxide, which is ultimately they causes the vasodilation, ultimately leading to the release of the excess heat from the body. And so we see that the Haryana is producing more Haryana in the level of enos is higher in case of Haryana. So the Haryana shows the better adaptation capability by releasing excess heat from the body. So from these observations, we see that the stress markers were well expressed in case of Haryana, showing that Haryana, which is an indigenous breed, it is more thermo-adaptable and it is more thermotolerant to high increasing temperature, global increasing temperature. And the level of the expression of genes such as heat shock protein, interleukin, and endothelial nitric oxide synthesis shows that Haryana is a better thermo-adaptable in case of higher ambient temperature as compared to crossbred animal. Hence, it is very important to conserve the local breed to maintain both production and the productivity of the animal during thermal stress period. So it can be concluded that the dynamic physiological and hormonal changes are favoring the acclimation to heat stress in case of Haryana cattle breed more as compared to the crossbred animal. So thank you so much, sir.
Thank you, Dr. Lipika Sharma. Now it is open for discussion. Respected chairman and co-chairman, please, if any participant member has queries, please proceed.
Dr. Lipika, please tell about what is the level of exotic inheritance in Brindavani cattle?
Sir, Brindavani cattle is basically a cross between Holstein Friesian, Brown Swiss, and Jersey, and the indigenous group is Haryana, and it has 75% 75% inheritance from exotic animals, sir.
Okay, have you estimated, because there will be further changes, or it may be moved towards the purebred condition, or the level of exotic inheritance will be declined?
Sir, I'm not getting the question, sir. Sorry, sir. Sir, please repeat the question, sir.
The level of exotic inheritance is maintained throughout your study or in previous study? That is 75/25 ratio?
Yes, sir. Yes, sir. It was maintained, sir. Throughout the study, sir, it was properly maintained, sir.
Okay. Okay.
Respected co-chairman, Dr. Satinder sir.
The presentation is clear. No question.
Okay,
Thank
Dr.
you so
Lipika
much.
Sharma, I have one question.
Yes, sir.
You have mentioned here the thermo-adaptability between indigenous and crossbred cattle.
Yes, sir.
So the parameter you have decided is the environment between the indigenous and crossbred cattle, laboratory environment is the same for the parameter? Because you are analyzing the thermo-adaptability on the basis of biomarker, like gene expression markers. So gene expression marker, gene will express when the animals become in more stress. So how you have decided the stress in the control environment or in your semi-control environment in your IVRI?
Sir, we kept the animal for 7 days, sir. That is the pre-exposure
7 days
period, sir.
Where you have kept? In
Sir,
7
in the
days,
normal, sir, in the normal, yes, sir, in the normal
normal
condition. Yes, sir.
So the
And
so
After that, we expose it for 49 days, sir. Again, after 49 days, we have kept, sir, after exposure period sample, sir. That is for, sir, 7 days sample. After 49 days, we have taken the sample for, sir, 7 days, sir.
So no,
Recovery
stress,
sample, sir.
what is the stress parameter you have decided for thermoadaptability? Means temperature related only?
Sir, we have seen several parameters, like, sir, we have gone for, sir, THI sir, examination, and also physiological, other biochemical parameters we have examined, sir. So we have, I haven't given it in the presentation, but we have recorded the feed intake, water intake, sir.
Okay. The shed in the IURI, they are made up of what the temperature variation in your shed is. Suppose 40 degree or 42 degree or 45 degree temperature degree centigrade is in environment. So below shed, what is the temperature?
Sir,
what is the difference between temperature open and in your shed?
Sir, it varies actually. Sir, the time we did, sir, the normal temperature at we have exposed it to 38, but before 38, sir, it was lesser than 38, sir. And also, sir, recovery period sample, it was lesser than 38 degrees, sir. So that we have considered to compare this, how animals, sir, normal physiological biochemical hormonal and gene expression marker expresses during 38 degrees Celsius to see that we have, sir, considered that the program, sir.
Okay, so it is not fully controlled environment actually, है ना? It is between communicating environment between outside and your shed, correctly?
Sir, during exposure, we control it 38 degrees Celsius, sir. But while taking, sir, pre-exposure and after exposure period, that was in, sir, safe, sir.
Okay. Thank you, Lipika.
Okay, sir. Thank you so much, sir.
Dr. Bhuri Singh, are you there?
Sir, please allow me to, sir, stop sharing the screen, sir.
We have allowed. You may leave.
Thank you, sir. Thank you.
Dr. Bhuri Singh.
Yes, sir.
Please proceed.
Okay,
make it
sir.
fast. This is last presentation.
Hello. Good evening, sir.
Good evening.
Sir, this is Ajay Kushwa, serial number 4. I joined from the agriculture presentation. So I left here my presentation. Can I present here?
Okay, you wait. Dr. Bhuri Singh, please share your slide. Okay. And after that, Mr. Ajay will present. Dr. Bhuri, please make it fast. Don't be late. Dr. Bhuri, please. Are you able to hear? Dr. Bhuri? Hello. Dr. Bhuri. Okay.
Hello. Sir, are you audible?
Yeah. Please proceed.
Good evening. Myself, Dr. Bhuri Singh, Assistant Professor, Genetics and Plant Breeding, Agricultural University, Kota. I am delivering on my poster presentation on topic multivariate analysis for grain yield and its contributing trait in wheat under southeastern humid plain of Rajasthan. The objective of my study at present, increasing bird population argues for provision of enough food to human and human races, and wheat is the major versatile food and fodder crops contributing importantly to nutrient supply of the global population. At present scenario, it is an urgent need to develop climate resilience variety, which is an economically and sustainable option via plant breeding approaches. Therefore, the present study was focused on the assessing the genetic diversity and explore the impact of trait that promote increased diversity among the cultivars. 42 cultivars of wheat were selected and evaluated in a randomized block design with 4 replication during Rabi season 2023 and 40. And the observations were taken on 5 randomly selected plants from each cultivar and each replication, eliminating border and unhealthy plant or the 12 trait, such as days to 50% flowering, maturity, number of tillers per plants, plant height, spike length, biological yield per plant, number of grains per spike, number of spikelet per spike, number of grains per spikelet, test weight, grain yield per plant, and harvest index. The replication wise recorded mean data of each cultivar for each trait has been subject to multivariate analysis. Principal component analysis computed following row 1964 as linear combination of optimally weighted observation variables and analysis of data by graph software and k-mean clustering analysis. One can customize on number of cluster to be formed, thus meaning validation of results are possible and grouping of the cultivar by OP state results. The existence of significant differences were observed among the cultivars for all the traits under study, indicating the presence of considerable amount of genetic variability in the cultivar. A wide range of variation were observed among the cultivars for most of the traits. The PCV and the phenotypic coefficient of variance values for most of the trait were closely matched to the corresponding genotypic coefficient of values. High heritability combined with genetic advance over mean was observed for the traits such as biological yield per plant, harvest index, and grain yield per plants. In principal component analysis, 4 principal components with eigenvalue greater than 1 contributing 69.56% of the total variability among 42 cultivar studies for 12 traits and 5 clusters were formed from 42 cultivar. Cluster 4th has the highest number of 14 cultivar, followed by cluster 2nd, and cluster 1st was solitary with 1 cultivars, and the cultivars 1st and the cluster 1st and 4th had the greatest inter cluster distance of 3.42%, indicating that the cultivar between each pair of cluster were more diverse based on the overall appearances and per se performance of the cultivar. Cluster 4 means cultivars Raj 4238, Raj 3765, Raj 4037, and
Please conclude.
2338, GW 336, and SD 2278, and GW 190, Sujata SD 2967, and SD 2781, and UP 319, and PBW 226, and SD 2189, and DVW 16. And GW 053 in cluster 1s were found more desirables and diverse. And conclusion, the selected cultivar can be employed in hybridization program and to produce better transcriptive segregant, offering a high potential for yield in subsequent generation.
Okay.
Thank you.
Okay, thanks, Dr. Bhuri. It is open for discussion, respected chairman and co-chairman. Please proceed for questions.
Dr. Bhuri Singh, you have checked your name is included in this session or in
Yes.
agriculture session?
Yes,
Because
sir.
this presentation is totally related with agricultural group.
Yes, sir. Yes, sir.
Okay, so that's why your name is not included in this veterinary session group. So
Okay,
please confirm about this.
Okay, okay, sir.
Okay.
Okay, sir.
Okay. Thank you.
Thank you, Dr. Bhuri Singh.
Okay,
Now next and the last is Mr. Ajay.
Yes, sir. I am here. I'm present.
अरे यार इसमें है ही नहीं वेटरनरी
My my presentation is seen, sir. I'm audible.
Yeah.
Yeah. Okay. Good evening, present here all. I am Ajay Kushwa, research scholar from Dalbagh Educational Institute, Agra. I am presenting a farm producer organization as an engine of rural development inside from the Sahaj milk producer companies. So, what is Sahaj milk? What is milk producer companies? A producer company is a business enterprises run on the basis of the mutual assistance principles, which are broadly similar to the cooperative principles adopted by the international cooperative alliances and cooperatives. A producer company combines with the institutional and ideological strength of the cooperatives, ownership limited and to users, limited interest to share, no trading of share, pathogens not capital based, with the flexibility and autonomy available under the company's law. So empowering the dairy farmers, transforming the livelihood by bringing dairy farmers together on their own enterprises as a producers organization, establishing direct milk procurement facilities and the reaching of the aggregate flood drought process, providing a market access twice in a day, and providing complete digital access to the dairy farmers through the mobile applications that hold information about the transaction with the MPOs, and enhancing the productivity of milch animals by the providing the dairy farmers access to the artificial inseminations. So this is the current status of MPOs in India. Basically, it covers a 10 state in India, and number of the MPO in India is 22, uh, with uh, with the 8 lakh 72 farmers. Most of the members of 71% of the farmers is women. Generally, 15 MPOs is running by the women, and these are the productivity enhancement services given by these companies. The current status of the MPOs in Uttar Pradesh. Currently, 6 MPOs operationalized in Uttar Pradesh. Three were the run by the women. The, uh, Now coming to the Sahaj, overview the Sahaj of the milk producer company. The company was formed in 17 October 2014. The alternative of the company started from the 12th of December 14th. The meaning of the Sahaj is neutral, simple, and spotless. So this is the operational area of these company. Basically, zone 1 is Agra, Firozabad, Etawah, Mainpuri. Zone 2 is Aligarh, Etah, Badaयूं, Moradabad, and Kannauj and Kanpur, Bihar, etc. So these are the services provided by the companies to the animals and farmers. First one is a balanced ration program to the enhance the productivity and the milk production of the cattle. The second one is artificial insemination. Company providing the doorstep AI services as a engaging qualified with a trained mobile AI technicians in this project area, enhance the productivity of the cattle. The third one is a fodder development program. The green fodder production enhancement, the improved fodder seeds productions program, silage making demonstration, and demonstrating revegetation of the common grazing lands. The features provided for the farmers is for the producers producers awareness program and a clean milk productions and the Women's Awareness Program organized, and the Rural Youth Awareness Program. The methodology are used for the resource collection of the data. Is primary data was collected from the farmers by the survey and the interview, and the secondary data was collected from the company's annual report. The sample size is generally size milk producers organization and their members, the farmers. So the primary survey is conducted with the 39 members with the all age group below 25 to the or to above 50. They are associate with the company as a dairy farmer, as a milk collector, and as a worker and a staff of the company. So from all these respondent, 58% of the respondent think about emphasis are providing the employment of the rural youth, and the 70% of the this showing the increase of the family income of the members of these employees. The women empowerment, 80% of the respondents give the positive feel for the women empowerment. MPCs provide the 38% of the women employment, 22% of the women financially independence, and the 17% of the women encourage the leadership. They are running the various milk producing organization in India. And the next one is 66% of the respondents showing the MPCs is supporting the community development programs by the supporting to the students program and etc. And this is a benefit experience by the farmers, just like veterinarian and technical support by the companies, better pricing of the milk, steady income, and community development program by the company. And this is the secondary data from the company. They are collecting the last year 5.37 lakh liter milk in a single day with the 3,110 cooling booth of the of various villages. These are total members is currently above the 1.10 lakh over the, and the 50% above the 52% of the members are women in this company. Simply artificial inseminations services given by the company to the farmers is over the 6,47,000. Now the training and the awareness program of the for the farmers given by the company. They provide the training and the awareness program to the development of the farmers and the women and the rural youth. Student training program as you can see, provided by the growth of the student as an entrepreneur in the village level.
Please focus on your research plan.
Yes, sir. I'm
Sir, you have mentioned the about the Sahaj Dairy Cooperative.
Yeah. Yes, sir.
Okay. This is also not required farmers feedback and such things.
No. Sir, my research is overall community development and the women empowerment by the company.
From the data from the 2015 to 23-24.
Yes, sir. Previous year.
This is the company profile. This is not the part of your research.
Sir, these data is collected from the primary survey of the interview of the farmers.
Okay.
Right? Okay.
You should focus on your research part.
Yeah, I am also doing the research with the socio-economic empowerment or in the women empowerment of these the farmers by these companies.
Okay, this one is okay. Wage growth and women empowerment.
Okay, now to the conclusion last. In the conclusion, the Sag milk producers company serve to the poetal example of the how farm producers organization can drive the rural development of empower the local communities, established with the vision of the creating the sustainability and equitable dairy industry. Sag has successfully engaged with the numerous farmers, thereby the facilitating direct access to the market and the eliminating middlemen. This model has not only enhanced the financial stability of its members, but has also inspired social changes by the focusing on the women and the youth empowerment through the awareness programs. The transformative impact of the SAS is evident testimonies of these of farmers who have experienced significant increase their income, improved educational opportunity and their children, and sense the security of their future. By forecasting a culture of the corporation with the sharing knowledge says, exemplify how aligning the local wisdom with the professional expertise can unlock the potential of the rural populations. Thank you, sir. These are the references.
Thank you, Mr. Ajay Kushwaha.
Yes, sir.
I request to Dr. D.N. Singh, sir, respected chairman, and Satyendra sir, Dr. Satyendra sir, respected co-chairman, may ask questions. Please, sir.
Please show about what is the adoption level among the farmer by providing the various components like training, extension services, because you have not mentioned what is the adoption level of your selected farmers.
So adoption level is generally farmers got training by the companies, and the improvement.
It should be the part of your research area.
Yes, sir. Yes, sir. It is my part of my research area. I'm doing research on the social
You have
economic.
only mentioned the age group of the farmers.
Yes,
You
sir.
have not conducted any survey prior to conduction of the experiment, and after conduction of experiment or after providing the various training or veterinary education like that. So,
No,
what is the overall impact of this organization or your study among the farmers?
yes, sir. So generally, the companies are providing a various awareness program and a training program for the farmers, and they.
So you have not mentioned now what is the impact of awareness program among the farmers.
Okay, this is a part of the research, and I will.
You have only focused about the details of cooperatives.
Women empowerment and basically improvement of their socio-economic conditions.
Okay, by the way, presentation was very nice.
Okay, thank you, sir.
Okay.
Thank you.
Any more questions from? Yeah, that's it.
Actually, presentation is good, and it's also they have selected good area in Uttar Pradesh, that is Agra and around, and Sahaj is also a good impact in this area. But sir, presentation knowledge adoption, that was should be covered. Knowledge enhancement, it should be covered. It should be reflected. Means directly you are putting result. Means how farmer perceived about your technology, that should be included.
Okay, I will improve it.
And before start of work and after at present, what is the differences between production, socioeconomic status, and even you can compare with the educational background and socioeconomic background, how it is differing.
Okay, sir. I will try to control all this.
Anyway, good presentation.
Thank you, sir.
And congratulations.
Thank you so much.
You have taken too much pain to collect the primary data and secondary data and compiling the information. Put some statistical tool for better
Sir,
better results and conclusions.
I am finding the more data and the more ground data for my PhD finalization. So I show you a small sample for this. Yeah, how the companies, how the milk producers organizations are supporting to the farmers at the ground level, just because they are collecting the milk on the grounds, the doorstep of the farmers by the pooling booth in the village level, and they are supporting directly to the farmers. Recent years, NDDB's program, I think you listen it, NDS National Dairy Services is directly promoting to the farmers with these all organizations, how they are growing, and how women, almost 15 MPCs are running by the women last 10 years. as we can show, the last 10 years 200%, almost 200% financial growth with the all MPCs showing, just like Kasi and Kasi from Banaras is showing a rapid growth just because of the basic member of the just because of the members of these MPCs is are women, and also employees also are women. They are running the cooperative societies. Thank you, sir.
Thank you.
Thank you, sir.
Okay, thank you, Mr. Ajay. You may leave now.
Thank you, sir. Thank you so much.
Now I hand over to Dr. Dinesh, sir. Please, sir, proceed further.
Thank you, one and all. So today is the end of technical session 2, veterinary and fisheries sciences. So I requested all the participants kindly leave the group except the panel member. So I think all participants are left the group.
हो गया दिनेश सर?
हाँ हो गया.
लगता है बस सारे चले गए
बचे हैं.
तो नमस्ते सर.
आज तो
नमस्कार.
आज तो बहुत ज्यादा हो गया, हम लोग तो 3.5 घंटा होने वाले.
आपने इन 25-30 जनों को भी एग्रीकल्चर का चल रहा है, आपको आवाज आ रही होगी. वो 60 जने अभी 25-30 में भी देखो बराबर से चल रहे हैं. उनके भी एक दो बच्चे
डिस्कशन ज्यादा हुआ.
अब आज कितने हुए?
आज एक्चुअली ऑन रिकॉर्ड जितना है उससे ज्यादा हो गया.
2, 3, 4.
तो इन्हीं में से
एक्चुअली 2
कल अनाउंस
पोस्टर
करेंगे ना,
एक्स्ट्रा है.
तो कल अपन अनाउंस करेंगे.
हाँ.
तो इनमें से
सर 10 ओरल हो गए हैं. 10 लोग ओरल हैं इसमें. 1, 2, 3, 4, 5, 6 लोग पोस्टर प्रेजेंटेशन किए.
6 पोस्टर
16 टोटल.
16 टोटल आज.
और जिसमें से डॉक्टर जसमीत कुमार है वो बीच में लेफ्ट हो गए थे, तो उसके बाद उनका हाफ ही प्रेजेंटेशन हो पाया.
अच्छा मेहता सर, ये बताइए जो एग्रीकल्चर में भी प्रेजेंट किया है और हमारे यहाँ भी कर दिया है, उसका क्या करेंगे?
सर मुझे आवाज कम आ रही है थोड़ी.
नहीं, वो एक एग्रीकल्चर वाला भी तुमने प्रेजेंट किया ना इसमें.
1 मिनट में जुड़ता हूँ सर, जुड़ता हूँ. 1 मिनट रुकिए.
अरुण है शायद. अरुण.
हाँ बोलिए.
भूरी सिंह.
हाँ, भूरी सिंह करके है.
एक
म्यूटेड.
ठीक है सर, अब फाइनल कर ले.
जी सर.
हाँ फाइनल कर लेते हैं सर. आज का सर टोटल जो है, टोटल प्रेजेंटेशन जो ओरल है कल और आज का मिला के 16 हुआ. जो 16 2 5 15 हो गया सर. कल 5 था और आज 10 था. 15 हो गया आपका ओरल और पोस्टर में 6 हो गया सर.
सर हम दिनेश सर हम फोटो खींच के आपको भेजे हैं दिनेश सर, उसमें सारा समरी है.
हाँ देख लिए. तो सर बताइए सर रिकमेंडेशन.
मेरा रिकमेंडेशन एक्चुअली मुझे सबसे अच्छा प्रेजेंटेशन लगा रविंद्र कुमार यादव का. ही हैज टेकन सिंपल टॉपिक और उसका एनालिसिस जो है बहुत ही अच्छी तरह से किया. एलपीटी सर लाइव ये क्या बोले
टेक्नोलॉजी.
जी नहीं, हम ऐसे ही बोल रहे थे कि जिसने एग्रीकल्चर में भी प्रेजेंटेशन कर दिया, हमारे यहाँ भी प्रेजेंटेशन कर दिया, उसको कहाँ काउंट करेंगे?
नहीं, अगर वो एग्रीकल्चर का है तो उसको उधर शिफ्ट कर दो.
हाँ उसको भेज दिया
कौन
उधर.
है?
एक
क्या नाम है?
भूरी सिंह था, भूरी सिंह।
हाँ, मुझे डाउट था, वो कोटा से हस्बैंड वाइफ दोनों, तो
हाँ।
वो उनकी वाइफ ने तो उधर ही कर दिया विवेचना ने, और इसने, मैं वही सोचा था इधर क्यों ज्वाइन किए हुए हैं, उनको उधर ही शिफ्ट कर देंगे, कोई दिक्कत नहीं है।
बहुत देर तक हम लोग सोचते रहे, उन्होंने बड़ा लंबा प्रेजेंटेशन दिया पोस्टर का, लास्ट में हम लोग यही बोले यार समझ में नहीं आया क्यों आप इधर आ गए।
सच में उनको पहले ही बता देते या तो स्टार्टिंग में।
अरे हम लोग इंतजार कर रहे थे, कुछ खेतों में कीड़े वाले भी थे ना।
अच्छा,
हम सोचे कि अब
सर जी सर।
जी सर, रविंदर कुमार यादव सही है सर, सत्येंद्र सर मतलब बोल रहे हैं, मैंने भी पॉइंट आउट किया था इसका, तो इसका
एक्चुअली
हमने मुझे बताइए सर कि कल के सेशन डे 1 के अलग करें कि डे 2 के दोनों का अलग करें कि एक साथ मिला के दे अवार्ड अपन?
एक सर अलग भी कर सकते हैं, साथ में भी दे सकते हैं। कल हमारे यहाँ प्रेजेंटेशन बहुत कम थे। और कुछ हमें अच्छे प्रेजेंटेशन लगे लेकिन उनके इम्प्लीकेशंस ज्यादा नहीं, तो जिसके कारण आज एक प्रेजेंटेशन जो था सिंपल टॉपिक बच्चे ने लिया है, ये लीची पल्प और स्वीट पोटैटो, और फिर इसका जो है कि उसने योगर्ट बनाया और उसको एकदम रिसेंट टेक्निक से सरफेस एनालिसिस जो भी मेथड्स हैं, तो वो बड़ा इंटरेस्टिंग प्रेजेंटेशन रहा। और देखा जाए तो हमें लग रहा है कि वो रिसेंटली पेटेंट भी इसको करने जा रहा है, तो ये हमारे लिए भी अच्छा रहेगा कि हम उसको रिकमेंड करें एज ए फर्स्ट।
बहुत बढ़िया
जी।
तो ऑल डिसीजन आप ही का है सर, आप ही अपने हिसाब से बेस्ट सेकंड थर्ड कर दी। अब वो डे 1 डे 2 करें कि ये एक साथ करें, ये एक बार कैसे करें इसको?
अगर डे 1 डे 2 करेंगे तो हमारे
कल तो
लिए
5
अच्छा
ही थे ना
होगा।
शायद,
5 या कल
नहीं 5
कल
अपन
5 ही
करेंगे
थे।
तो 3 के बाद चौथे, हाँ कल 5 में से 3 को कर दें, कहने का मतलब ये है ना ऐसा।
हाँ।
कल 3 हो जाएगा, आज 3 हो जाए तो फिर 15 में से 6 हो गया, ऑलमोस्ट
15 में 6।
आप तो ठीक है, अगर एनकरेज करना है तो ऐसा किया जा सकता है, क्योंकि इसमें एग्रीकल्चर में भी शायद यही करना पड़ेगा ताकि ज्यादा लोगों को एनकरेज हो कि भाई मेरे को भी मिला है।
जी।
और नहीं तो वो कल वाले कह देंगे हमको तो जिन्होंने कल दिया उनके तो किसी के नाम ही नहीं आए, ये भी हो सकता है।
नहीं नाम तो आ रहे हैं साहब, हम लोगों ने सबको कंसीडर किया है और क्रिटिकली देखा है।
कल वाले के अगर अपने कल आज के मिला के देंगे सब।
ऐसा करते हैं ना कि जैसे फर्स्ट सेकंड थर्ड में, उसके बाद जो है थर्ड में कंसोलेशन प्राइज वो कर सकते हैं।
ये भी होता है, ये भी होता है, ये सही है,
कंसोलेशन कर सकते हैं।
हाँ, कंसोलेशन 2 प्राइज।
हाँ 2 कर देते हैं कंसोलेशन, 5 टोटल दे देते हैं 15 में से। इधर पोस्टर चाहिए तो उसमें से फर्स्ट सेकंड थर्ड हो जाएगा।
हाँ ये भी हो सकता है कि दोनों डेज को क्लब करके सेकेंड थर्ड या फर्स्ट पे जो है दो को जो है रख सकते हैं, दोनों को जो है एक साथ क्लब किया जा सकता है।
क्या एक सेकंड।
ठीक है सर, अपना लोग डिसाइड कीजिए सर, हो गया, कोई दिक्कत नहीं है। फर्स्ट जो है कोई दिक्कत नहीं है, 5 कर सकते हैं।
फर्स्ट रविंदर कुमार यादव सर ने बताया। सेकंड
सेकंड में कौन?
सर्कल कल का एक अरुण कुमार का था, अरुण कुमार या अरुण कुमार ऐसे।
हाँ आर अरुण कुमार है।
आर अरुण कुमार है। और बबीता का था, तो बबीता का उतना सिग्निफिकेंट, हाँ लेकिन ये था सस्टेनेबिलिटी रिलेटेड था, उसने इंटेरिक मीथेन इमिशन पे टॉक दिया था, तो उसको भी अपन कंसीडर कर सकते हैं। और एक जो है फिशरीज से अपन ले सकते हैं।
डॉक्टर सरली सर फिशरीज का उसका प्रेजेंटेशन कंटेंट
फिशरीज पे जयाश्री का और मधु दोनों का प्रेजेंटेशन अच्छा था। जयाश्री का जो प्रेजेंटेशन था वो सीवीड से कैसे, सीवीड जो है कि समुद्री,
जी सर, जयश्री का भी सस्टेनेबल से रिलेटेड था सर प्रेजेंटेशन।
हाँ, सस्टेनेबल से रिलेटेड था। अच्छा प्रेजेंटेशन कि कैसे आप सोर्स है उससे लेदर अपन बना सकते हैं सीवीड से। इनोवेटिव थॉट है।
करेक्ट सर। और टाइम भी इसका सही था सर, इसने विद इन
जी
5 मिनट में अपना टाइम भी, पूरा प्रेजेंटेशन में भी टाइम भी करेक्ट लिया है।
चलिए ओके।
और सर
तो जी,
जैसे सर रविंद्र कुमार यादव और अरुण कुमार को फर्स्ट पे रखे हैं, दोनों को
फर्स्ट पे नहीं रख सकते हैं। अरुण कुमार टाइमली आया नहीं था, रविंद्र कुमार ने टाइमली फिनिश किया, टाइमली
अच्छा, सेकंड में सर, अच्छा सेकंड में सर हो गया, फर्स्ट में रविंद्र का, थर्ड में सर, थर्ड में 2 दे दीजिए, कोई दिक्कत नहीं है।
थर्ड में भी 2 हो जाए।
एक जयश्री को दे देते हैं
जी,
अरुण कुमार को देखिए थर्ड में ही ले आइए, जयश्री को सेकंड।
अच्छा ठीक
उससे
सर,
ये होगा कि थोड़ा सा ये, एक तो ये जयश्री की है, इनको साउथ में भी एक्सपोजर चाहिए और इनोवेटिव वर्क की है फिशरीज।
हाँ सर, हाँ थे इसका,
तो इसको सेकंड पे ले लें और थर्ड पे 2 को ले लें।
हाँ तो एक सार्ली हो गया सर थर्ड पे और एक अरुण कुमार, ठीक है।
सार्ली का लास्ट का प्रेजेंटेशन।
जी सर, डॉक्टर शार्लिन का फिश से रिलेटेड था सर, फिशरीज के ऊपर था।
जो अभी मेराइन वाला जो किया, वो गंगेटिक वाला जो किया है सर,
वैल्यू का पूछा था चेयरमैन सर ने, न्यूट्रिशन वैल्यू के बारे में पूछा था।
ज्यादा कुछ खास तो नहीं था उसका प्रेजेंटेशन, लेकिन दे सकते हैं। ठीक है, अपन सेकंड थर्ड में 2-2 कर दें, खत्म करें,
टोटल 5 हो जाएगा।
हाँ टोटल 5 हो जाएगा।
सेकंड में और किसको रखना है सर, बताइए फिर।
सेकंड में एक दीप्ति का कल का प्रेजेंटेशन है और आज का एक डीएनए आरएनए था।
हाँ वो लड़का जो दिया था वहां पे एनडीआरआई का,
एनडीआरआई का।
हाँ
सर ये मिसेस दीप्ति का जो प्रेजेंटेशन था सर, जो मॉस्किटो के ऊपर था,
दीप्ति का भी प्रेजेंटेशन अच्छा था, लेकिन उसका इम्प्लीकेशन मुझे समझ में नहीं आया कि कैसे जो मिरर रिपीट्स है उसको साइलेंट करके ऐसा बना देगी कि मॉस्किटो काटेगा भी तो आदमी भी मर नहीं।
सर वो एक्चुअली क्या है ना, बाहर जो कुछ फॉरेन कंट्रीज हैं तो पॉपुलेशन क्रिएट कर रहे हैं, ब्रीडिंग करा रहे हैं। जैसे बाहर जो वाइल्ड मॉस्किटोज है उसकी ब्रीडिंग जो लैब में क्रिएट हो रहा है मॉस्किटो, उसकी ब्रीडिंग करा रहे हैं। तो इससे क्या होगा कि वो जो क्रॉस ब्रीड हो जाएगा।
हाँ,
तो
दीप्ति को तो मैंने भी रखा है, दीप्ति को भी दे सकते हैं।
जी सर।
जी,
तो दीप्ति को सर कौन सा, सेकंड में दे दे या थर्ड में?
दीप्ति को सेकंड में दे दीजिए, अरुण कुमार को थर्ड में दे दीजिए।
ठीक है सर, तो 5 हो गया, रविंद्र कुमार का फर्स्ट
5
में
हो गया।
हाँ, सेकंड गया श्री का और दीप्ति का, और थर्ड में अरुण कुमार और सरली का। ओके, पोस्टर में सर 6 में से
पोस्टर में लिपिका शर्मा का पोस्टर आप लोग कैसा लगा?
अच्छा था सर, अच्छा था सर, सही आइडेंटिफाई कर रहे हैं सर, ये अच्छा था सर।
और ये इंफ्रारेड थर्मोग्राफी रियल टाइम रिजल्ट देगा।
भगवती का सर, भगवती का
जो भगवती का
नया इमेजिंग टेक्निक,
अच्छा प्रेजेंटेशन था।
और ये तब कैनवास में काम चल रहा है अभी खाली।
नाम क्या है? नाम
कहीं है नहीं है।
नाम लिपिका शर्मा, भगवती एम और एक पूजा गिल का था। हिस्टोलॉजी वगैरह अच्छे बनाए थे उसमें, हालांकि मुझे ज्यादा समझ में आया नहीं
सर। वो एक्चुअली एलिफेंट में था, वो पोस्टमार्टम रिपोर्ट थी, एक केस रिपोर्ट है
जी।
और उसमें क्या है कि जब एनिमल्स बहुत ही जेनेटिक हो जाता है, बुढ़ा हो जाता है, तो उसकी केस स्टडी की है, लेकिन उसका आउटपुट क्या है सर?
आउटपुट क्या है, इम्प्लीकेशंस वगैरह
बुढ़ापे में अगर पोस्टमार्टम करे तो ऐसा कोई आउटब्रेक का उस तरह का कोई वो सिग्निफिकेंट नहीं बनता है कि हम उससे कुछ रिलेटेड इन्फॉर्मेशन तुरंत दे सकें। तो
लिपिका, भगवती और फिर तो एक गोल्ड माइन आपको देना पड़ेगा काउडंग पे जो काम की है
उसका, तो अच्छा था।
विकसित भारत में हम लोग उसको कंसीडर कर रहे हैं। रूरल लाइवलीहुड के लिए।
इको फ्रेंडली था सर इसका अफराज का, मिसिस अफराज
अफरा
का
का।
इको फ्रेंडली जो पता जो इसके ऊपर था आपका, जो आपने डिस्कशन किया
जी
था
जी।
पूरा प्रैक्टिकली
उसका
एप्लीकेबल, प्रैक्टिकलिटी भी देखें तो उसकी पोस्टर की थी थोड़ी, तो थर्ड में रख सकते हैं सर अगर चाहेंगे।
हाँ, उसको थर्ड में रख ले। डॉक्टर
ऐसा करते
लिपिका
हैं ना सर,
भगवती जी।
सर थर्ड में 2 का कर लेते हैं और फर्स्ट सेकंड रख
हाँ।
लेते हैं। ठीक है।
बेटा ऐसा भी तो होता है, फर्स्ट एक और सेकंड थर्ड दो भी तो होते हैं।
हाँ हाँ, वो कर
नहीं,
लिए हैं
वो
ओरल
इसमें
में।
कर
वो
रहे हैं,
कश्मीर में लीक किया
प्रेजेंटेशन
था।
में वो कर रहे हैं, पोस्टर ओरल प्रेजेंटेशन में 5 कर रहे हैं हम लोग, और पोस्टर में अभी डिसाइड कर रहे हैं।
वो कल एक पूजा तंबोली मैडम का कैसा था उनका?
पूजा तंबोली
कल आप कह रहे थे, मतलब इसलिए पूछा मैंने।
कल का
वो ब्रेजिंग बिहेवियर बताई थी
नहीं
कैमरा
नहीं,
लगा के
आज
वहाँ।
आज के केस में वो कुछ खास नहीं रहा। आज उससे अच्छे
सही बात
प्रेजेंटेशन
है,
रहे, ज्यादा प्रेजेंटेशन रहे
बात है।
जी।
भगवती का भी टाइम एकदम करेक्ट था सर, अफराज का भी टाइम
हाँ
पोस्टर का सही था जो आपने
सर
सिलेक्ट किया है।
तो सर जो पोस्टर में जैसे कि लिपिका हो गया, भागवती हो गया, अफरा हो गया, डॉक्टर अफरा और पूजा गिल तक, किससे सर सीक्वेंसिंग रैंक बताइए फर्स्ट सेकंड थर्ड का।
एक मिनट पूजा गिल का
तो डॉक्टर लिपिका ने 5 मिनट से ज्यादा लिया था, 3 मिनट मतलब ज्यादा लिया था, और बाकी जो भागवती है और अफराज है इनका टाइम एकदम करेक्ट है, मैंने लिखा हुआ है पूरा, तो देख लीजिए। बाकी प्रेजेंटेशन तो डॉक्टर लिपिका का ही अच्छा था सबसे पोस्टर में,
हाँ
इंफॉर्मेशन भी अच्छी थी।
जी जी। अगर एक पॉइंट पे अपन करें तो भगवती को 1 कर सकते हैं।
सर जैसा आप ठीक समझे सर।
चलिए भगवती को 1 कर देते हैं, लिपिका को सेकंड कर देते हैं।
अच्छा, और थर्ड में डॉक्टर अफरा और पूजा का।
पूजा का कुछ सिग्निफिकेंट नहीं है, और रेगुलर एक्टिविटी है, पोस्टमार्टम वगैरह हिस्टोलॉजी करना। तो
और सर बताइए।
16 नंबर पे है आरती गुप्ता, इसका कुछ खास नहीं था, कड़कनाथ को जीरा खिला दी,
हाँ हाँ, नहीं नहीं नहीं, वो
माइक्रोमेस्ट्री
उसने
चेंज कर दी।
एफसीआर भी देखी नहीं है, वो इकोनॉमिक्स भी देखी नहीं है।
हाँ, तो अब यही तो बच गया, और कौन है पोस्टर में।
तो डॉक्टर शिव
सर
डॉक्टर
सर जो सत्येंद्र सर ने आइडेंटिफाई किया वो एकदम सर सही है, मतलब कि पॉइंट और नॉलेज वाइज टाइम वाइज मेरा एक वो ही था, तो मैंने बता दिया सर को, अगर कंसीडर करेंगे सर तो डिसाइड कर लेंगे।
अरे
सर
कैसा बोले।
जितने सर,
क्योंकि प्लस माइनस 1 मिनट मैंने दिया है कि अगर किसी का नेटवर्क कनेक्शन है तो वो दिया है हमने, तो वो करेक्ट करके ही 5 मिनट हो रहा है।
डॉक्टर दीप ने सर।
हाँ हाँ
हाँ सर, लिस्ट जो है पोस्टर में,
हाँ, पोस्टर में ठीक है, फर्स्ट में एक को, सेकेंड थर्ड में 2 को रखना है ना।
हाँ
तो दूसरा डॉक्टर शिव कोई है क्या पोस्टर में।
नहीं सर, मतलब पूजा गिल का बस वही था कि आउटपुट कुछ नहीं है सर, अब बुढ़ापे में अगर कोई
तो फर्स्ट सेकेंड थर्ड कर देते हैं, और क्या करना फिर।
हाँ सर, मुझे लगता है कि जरूरी नहीं है।
आरती गुप्ता का एक था, तो उसमें मुझे कुछ ज्यादा नहीं, जीरा खिला दो तो।
नहीं नहीं, ठीक है सर, कोई दिक्कत नहीं फिर।
ठीक है, 1 2 3 करके ही छोड़े।
ठीक सर, ठीक सर।
ओके
चेयरमैन सर। हेलो हेलो
वो
हेलो।
सर उनका कनेक्शन लॉस हो गया है।
अच्छा, इधर फोटो दिखा रहे हैं।
नहीं वो
ओके
चले
ओके,
गए सर वो, तो
थैंक
कनेक्शन
यू
दिख।
सर। एक्चुअली दिस सेशन इज वेरी हेक्टिक से शेड्यूल।
आ गए सर, आ
दीपनाथ
गए।
सर। हाँ 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 सर, We already finalized the 5 award nomination in oral category group and 3 from the poster category group. Is it okay, sir? 3, sir. 3 out of 6.
हाँ ठीक है, सही है.
So thank you, sir. Actually, this session is very hectic today. We are joined almost 3.5 hours. Continue the session. So thank you, Chairman sir. Chairman sir,
and thank you Satendra sir. Continuously सब करने के लिए.
He is including full time.
Yes sir, yes sir.
हो गया सर
3.5 घंटा.
फाइनल.
हाँ फाइनल हो गया है.
चलो बढ़िया बढ़िया बढ़िया.
हम आपको भेज देते हैं.
जी जी.
सर आपको बहुत.
दिनेश सर आप ही अनाउंस करो ना फिर कल, या कोई भी कर दे आप दोनों.
अच्छा ठीक है, हम कर देंगे.
स्टार्टिंग इसी से भाई, आपको ज्यादा बेस्ट पता है ना, तो आप लोग ही कर दे तो ज्यादा बेस्ट है.
ठीक है, हम कर देंगे अनाउंसमेंट. टाइम बता दीजिए
भाई. कल कॉमन लिंक रहेगा तो 2:30 पे अवार्ड की जो अनाउंसमेंट है तो यहीं से हो जाएगी. और क्या आप मिलके कर दें, फर्स्ट सेकंड थर्ड जो कोई पूछे उसका एक्सप्लेनेशन भी मांगे तो आप बेटर दे सकते हैं ना.
ठीक है.
कोई बेईमानी करेगी तो नाम बोल के चुप हो जाएगी. आप एक्सप्लेन भी कर सकते हो क्यों उनको किया, टाइमिंग और न्यू फाइंडिंग्स और क्या वगैरह. तो आई थिंक वो ज्यादा बेटर है.
वो सब लिखा हुआ है सर, कंटेंट वाइज, प्रेजेंटेशन वाइज, वो सारा लिखा हुआ है आज के प्रेजेंटेशन का. सर को भेजा भी है मैंने उसमें, जिसका
ठीक है जैसा भी होगा
वो किया हुआ है.
कल संडे भी कोई दिक्कत नहीं है. 2:30 पे और तुरंत हो जाएगा. 10 मिनट लगेंगे ज्यादा. और क्या होगा, 10-15 मिनट.
ठीक है सर, कोई दिक्कत नहीं है. थैंक यू सर. चेयरमैन सर, को चेयरमैन सर,
थैंक यू सर, ऑल ऑफ यू सर. बिल्कुल आपका पेशेंस. 4 घंटे हो गए. कितना टाइम हो गया? 4 घंटे हो गए.
4 घंटे हो गए.
थैंक यू दिनेश सर.
थैंक यू आप सभी को बहुत बहुत धन्यवाद. अब फिर अगली बार जब मुलाकात होगी तब हम फिर डिस्कस करेंगे.
बिल्कुल सर, ऑफलाइन में बुलाएंगे. अब ऑफलाइन में मिलेंगे सर.
जी, बहुत अच्छा अनुभव रहा. तरह तरह के प्रेजेंटेशन. ऐसा कभी कभी तो ऐसा हुआ कि लास्ट में वो सिग्निफिकेंट बात बता रहा है.
जी.
क्योंकि ऑनलाइन में थोड़ा दिक्कत होती है ना, जिसके कारण कई चीजें छूट जाती है.
कनेक्शन लॉस होता है, फिर शेयर नहीं हो पाता है. बहुत सारा इससे कम टाइम लगता है सर. ऑफलाइन प्रेजेंटेशन होता तो.
हाँ, ऑफलाइन में कम लगता है.
सर हिसार और गो आ जाइए आप इसी साल. अभी 2 और ऑफलाइन इवेंट.
ओके.
अगस्त में हिसार में, नवंबर में गोवा में. अब आपकी चॉइस आपकी है.
ठीक है मेहता साहब, बहुत बहुत धन्यवाद.
जी सर. ओके सर.
ओके, थैंक यू
सर. गुड नाइट सर.
गुड नाइट.
थैंक यू सर. गुड नाइट ऑल ऑफ यू.
गुड
नाइट सर.
गुड नाइट.
गुड नाइट.